My name is Asha, and I'm very grateful and honored to be here today. I'm an instructor at Dr. Vikram Khurana's laboratory at Brigham Women's Hospital Neurology. And today I'd like to talk to you a little bit about gene therapies for cerebrospinal ataxias and other disorders. I'm very grateful for your flexibility for me to be able to record this video a few days ahead. I'm actually going to step into the plane in a few hours to fly to Poland to visit my father and my 101-year-old grandfather. I haven't seen him in 3.5 years because of COVID, and I'm very grateful for the scientific discoveries that allowed me and my 4-year-old daughter to be fully vaccinated now so that we can actually go together and visit my grandfather in a very safe way. Today, I'd like to talk to you about gene therapies for neurodegenerative diseases and cerebrospinal ataxias. So I'm going to share my screen. And I've been working on gene therapies throughout my career. And it is actually a great pleasure to be here now at Neurology at Brigham Women's Hospital, because I feel the work that's been done and ongoing here is amazing and has such potential for transformative medicine and benefits of the patients. So it's a great place for me to be here and also share my vision with Dr. Vikram Khurana, where we're really trying to do research to benefit patients. So what is genetic disease and what is gene therapy? Genetic disease is actually very much implicated here through this Silva family tree. Basically, tremendous work to be done to actually put this together really shows that what genetic disease is, which is a very specific disease that's being passed on through generations due to a specific mutation in one gene. And so, and what is then gene therapy? Gene therapy is actually a strategy that hopefully can be actually beneficial to patients who carry such genetic diseases, because what it does is can target specifically that gene and maybe repair it. So there's actually a clinical trial ongoing that started earlier this year, which we're very excited about for SCA3. And this is a gene therapy clinical trial. I'll talk to you a little bit more about it later. But here, this is an implication of how this kind of works. It's basically that we will have a gene therapy design here through antisense oligonucleotides, so synthetic pieces of DNA that can bind to a gene, the SCA3 gene, the gene that carries the CAG expansion. And by binding, it actually, through its mechanism, what it can do is actually can degrade this gene. So this gene is not functional anymore. So then the CAG expansion that causes the disease is not present anymore in the body. So that's gene therapy. The idea is that it's targeted and it's specific to that gene to fix that gene in a way. There's been actually tremendous progress happening in a few years regarding gene therapies for incurable brain diseases. And in early years, we really focused on actually developing the tools and making good and safe tools. There have been some clinical trials, which some of them have not been successful, which then actually kind of helped us to revert and go back and improve our tools to really make sure that what we're doing is really benefiting the patients by targeting the right genes and being specific and safe about it. And then in the recent years, gene therapy in the form of antisense oligonucleotide, that's predicted here in this graph, has been very fruitful. And clinical trials have actually resulted in FDA approvals for multiple diseases and multiple treatments available now for either Duchenne muscular dystrophy or SMA or even specific subtypes of Button's disease. What's very excited to us, made us very excited, is that earlier this year, there was a clinical trial started for gene therapy for SCA3, and I'll tell you a little bit more about that later. So what is an ASO and how does it work? ASO is otherwise known as antisense oligonucleotide, is a synthetic DNA, piece of DNA. And what it does, it alters our gene expression. So every cell, as you know, has DNA, and then genes are being kind of made from that DNA. And so what ASOs can do, they can do two things. One of them is they can actually shut down our genes because they're specific they can shut down the gene that we want them to shut down. And so they do that by binding to the DNA and degrading that piece of DNA that's responsible for that gene. And so there's actually a few clinical trials ongoing with this kind of strategy of shut down, therefore lowering the levels of this gene, this protein that causes the disease. And those are for Huntington disease, SCA3, and ALS. A second strategy, it's actually a strategy that causes a gene is still being made, but that the mutation that's in the gene is being skipped. And what happens is that ASO here binds to this mutation, to this area where the mutation is, and that area can be then excised from the gene, and then the gene is still being made, but it's missing that piece of the mutation, therefore the mutation is not there anymore, so the gene is again normal, or as close to normal as possible. And so actually this strategy has been very successful for many diseases which are already on the market now for patients to use. And this is for SMA, for Duchenne muscular dystrophy, or Batten's disease. So why are ASOs so promising? They're very specific, so we can really target, but also they're actually very easy to manufacture. And the chemical moieties, which are around that piece of the synthetic DNA, actually have been recently tremendously improved to induce a very good absorption of this ASO into the cells, distribution throughout the body, and stability, and most important, safety as well. So that's why this field has been actually booming in recent years. So how is this gene therapy applied? It's actually very simple. For such kind of complex system, the application is actually very simple. It's applied through intrathecal injection into the lower back, in this kind of between spinal cord space. So we basically do an injection here of these compounds, the ASO compounds, and by using the body's own way, how the CSF, cerebral spinal fluid, goes between the spinal cord and the brain and goes into all the places, we actually now use the body's own way to deliver this drug throughout. So then basically this drug goes all the way from the spinal cord to the brain. And using the system, what we can then ensure is that this gene therapy, which needs to target all the cells in our central nervous system, can actually go into the space and do its job and bind to this mutated gene, in this case, for example, the SCA3. But there have been challenges. And in recent years, actually, we got some negative results from full clinical trials, which showed the importance of us understanding not only the tool and the therapy strategy, but also the mechanism of the disease. And so for a year and a half ago, there was a setback for Huntington disease trial, where initially very promising ASO trial actually showed that some patients who were taking this ASO treatment for a longer time were actually doing worse compared to the placebo, which was not great, of course, and showed that there's a problem with the safety. Also, recent trials for ALS, targeting two different mutations, two different genes, were actually halted because they didn't show much of benefit. And that actually recently, a few weeks ago, one of these trials actually showed that if they looked at the patients much longer after treatment and give them treatment for a longer time, they are now starting to see benefits, really outcome benefits. These patients doing much better. But the official trial was already halted because it didn't show any of this. And that actually tells us the trial design also needs to change, because these diseases the effects are so tremendous that perhaps the benefits need to take much longer to actually be seen. So we have a lot to do in our community and a lot to think about. And these trials really showed us that there's more to just targeting a gene. We also need to think about the mechanisms and the way how the body and the biology of the disease works. And so in our lab, together in collaboration with Dr. Timothy Yu, we have a program for polyglutamine disorders, where actually by building human stem cell models, we want to, of patients with these diseases, we want to undergo and determine the efficacies and safeties of ASO gene therapies. And so in a few minutes, I'll tell you a little bit about what we do and for one of these diseases. Basically, we have a program that targets CG expansion and dentatorubral pallidoluysian atrophy, otherwise known as DRPLA. And the goal here is basically to model the patient's disease in a dish, to then test the treatment before we try it in a clinic, to make sure that this treatment is actually a good strategy. What is DRPLA? DRPLA is a very rare genetic neurodegenerative disorder with a widespread central nervous pathology, so similar to SCA3. And similar to SCA3, it is also induced by an increased number of CG repeats, but in this case, in the gene called atrophin-1, ATN1. Any number of CGs above 48 will induce the disease. There's no disease-modifying treatment. This is a very rare, even ultra-rare disease. And the clinical outcomes show that the patients suffer from ataxia as well, but also very common epilepsy and at the end, dementia. We actually have one patient that we are designing gene therapy for, and his name is Tayson. Tayson is currently 14 years old, but he had shown a very early onset of the disease. And when we look at his clinical data, we actually, it's already started around 4 years of age. Tayson has 67 CG expansion repeats. And so similarly as in the SCA3 program, what we had done is we obtained Tayson's fibroblasts, and through the technology of reprogramming that awarded a Nobel Prize, we were able now to make induced pluripotent stem cells from Tayson. So these cells which we can then make into any cell that we want. And in our case, we're very interested in neurons and what happens with the neurons of Tayson's neurons, and what is the pathology of these neurons. And so we made Tayson's neurons in a dish in our lab. And so with this project, what we like to do is to validate and de-risk ASO gene therapy as a strategy for DRPLA. So like I mentioned, this is a collaboration between the ULab at Boston Children's Hospital, and they are actually, their part is to design ASOs through the stepwise screening strategy, and then design, screen, refine, and validate all of these ASO molecules. Just to give you an idea, basically, we actually have over 10,000 molecules that we start here at the beginning, the designing. This is on a computer, of course, and then we take the best candidates and go through with lab work, with cell validation assays, with in vivo actually toxicity assays as well in animals. We finally get to a couple dozens at the end of molecules from this 10,000 that we think are worth looking and pursuing as candidates for clinical. And then in our lab, at the Kurana lab at Brigham Women's Hospital, we are now testing and de-risking these ASOs as potential therapeutics. And so using Tayson's neurons, now by treating them with ASO, we have these treated neurons, which should potentially be responding as healthy neurons as well, if the gene therapy is successful. And so there's two goals of this, is basically to the idea with this ASO is to reverse the phenotypes observed in the dish, and then to confirm that these compounds are actually safe. So we can also do that now. We have systems to do that in the dish using cells of a patient to study how are they affected by this gene therapy and if this gene therapy is safe. So here I'd like to show you one of the things that we study and look at. Like I mentioned to you, DRPLA, patients with DRPLA suffer from severe epilepsy. And so one thing for us we're thinking about is, can we model this epilepsy in a dish? And then can we actually rescue it? If we can rescue and take away the epilepsy from our patients, that would improve their quality of life tremendously. And so we started working on this as preliminary data, but we started to look at neuronal activity in a dish, which can, in a way, be showing also how the neurons are active in the brain. And so here we have neurons, which are marked in this green, and we look at how are they active in a dish. So basically what we do is we look at them just under microscope, put them on there, and using this kind of green dye, they should start showing us, are they active or not? And as you can see, this is healthy neurons. You can see many of these blinking, and that actually shows that these neurons are firing, we call it. So they're active, and they're sometimes active together. Sometimes they're active just by themselves, blinking here, or one starts and the second goes, so they're connected. So that's how neuronal activity can be measured, by looking at these, the amount of blinking. And then when we look at these activities of our DRPLA patient neurons, we actually start noticing that there's different patterns here. And what you can see, these neurons blink a lot more, brighter, and much more together, or we call that synchronized. And so this was very exciting data for us. Again, this is kind of early days, but when we quantify these, we look at two different measures of quantification of this activity, either the amount of kind of how much do they fire, how bright these images are, or how often do they fire. And both of these measures that show neuronal activity were actually increased in our DRPLA patient neurons, which was very interesting to us. And even more interesting, when we treated these neurons now with ASO, a potential candidate ASO, we actually found that that decreased the level of activity of these neurons back to healthy subject levels, which is really promising to us. So, but what we need though is right now we're comparing these phenotypes to healthy subjects, cells that we got from other donations. But as we know, we all are different. All of us, our neurons may be doing different things. And so this is not an ideal comparison, because we cannot really say that this increased activity is really due to a mutation. And for that, we need gene-edited isogenic control cells, which means basically we take Tayson cells and we, through CRISPR-Cas9 gene editing, a technology that I know you've heard a little bit about, we can actually now correct the mutation and make these corrected lines, which should be healthy cells, but exactly with the same, and everything else in these cells is the same except for that mutation. And then if we're able to say that these cells are firing normally, we can say that this is really due to mutation. And second, what we can do with this gene-corrected line is that we can then compare, is our ASO as good or bringing this neuronal activity to good levels as this gene correction, which should be a gold standard for neuronal activity in this patient. And so I'd like to very much thank the Silva family here, because just a few weeks ago, I had received a Silva family Ataxia scholarship to actually be able to fund this isogenic control generation based on the work that we had done in this Cath3 program. So I'm really, really grateful for this, because it will really push our DRPLA program forward. So right now, I'd like to tell you a little bit about this Cath3 project, the clinical trial that actually has been started earlier this year. And just briefly, again, what this trial does is it targets using antisense oligonucleotide. It targets this Cath3 gene, which then degrades the gene levels, and so that the CG expansion, which normally is a cause of the disease, now it's not there anymore. And I'm showing you here some very preclinical data from a mouse model on this tool that is now in clinical trial. So what they did, they treated mice with ASO. They had either wild-type healthy mice or mice with SCA3, a model of SCA3. And either these wild-type mice are doing really well on the locomotion. So this is just measuring locomotion. They're just walking around being happy. Whereas the SCA3 mice in reds are having a lot less of activity, locomotion activity. And what is really remarkable is that with injecting the SCA3 mice, now with this antisense oligonucleotide, this gene therapy, they can bring back the locomotion to a level of a normal, healthy mouse. So this functional data is really amazing to see, and really shows potential of this gene therapy to be successful. And so, at least for the reversal, perhaps hopefully a reversal of pathology in a human in a clinical trial. And so here, this clinical trial was started earlier this year, and we're very hopeful to see some positive data soon on it. And now I'd like to give you an update of our progress on developing these SCA3 isogenic gene-corrected control cells. So as you know, Dr. Vikram Khurana was able to collect fibroblasts from your family, from the members of your family, from Mark and Veronica, and with their cells having either 62 or 65 CEG expansions. And we were able to generate iPS cells, and now we have them growing in our lab. And we sent these iPS cells to Harvard Stem Cell Institute core to be gene corrected. So as you can see here in the graphic, we are with this new technology that Harvard Stem Cell Core collaborated with us thoroughly, and we really got this now working pretty well, I think, in their hands, to actually decrease the number of CG repeats from 62, 65 to 10 CG repeats. So this way, the cell line should actually have a number of CG repeats that a normal healthy line does, with everything else being exactly the same in the cell, but this gene mutation is actually now being fixed, or gene corrected, we call it. And so the Harvard Stem Cell was able to generate these lines, and we have basic QC already on them being positive. And now we were able to send these lines to Garvan Institute in Australia for whole genome sequencing to confirm that these lines do indeed have this healthy amount of CAG expansions and the SCA3 gene, and also to make sure that the lines are clean. And so therefore there's no unwanted gene edit effects. So this is, we should be hearing soon from Garvan, and we're very excited to work with these lines to, one, really pinpoint the cellular pathology that is dependent and caused by the CG expansions, and two, to be able to test the ASOs that we had received from University of Michigan that Sharon was working on already, to be able to show that these ASOs indeed can bring actually the pathology and reverse the pathology back to the normal levels, that a line carrying the CG expansion of a healthy line derived from a patient originally is actually showing, so that this phenotype is truly a true rescue. With this, I'd like to thank everyone for their contributions to this work. It's been tremendous collaborative work from many different labs on the project of the DRPLA, and the funding on that through Cure DRPLA, and also now funding for us generating the iPS-derived lines from Silva Ataxia Foundation. I would also very much like to thank the Silva Ataxia Foundation and Silva family for supporting our work in our lab and at the Movement Disorder Division, and at the Brigham and Women's Hospital to really push this research forward for us to be able to expand the horizons and to help patients. Thank you again for your for acceptance of this unusual format of this talk, and I really wish you a wonderful meeting. Bye.